Hello, everyone. Today, we will introduce an article titled, Fast Bootstrap and Reliable Readout Using Hidden References for DNA Data Storage. Compared with the complex de novo assembly, resequencing uses a reference genome to enable efficient genome analysis. Inspired by resequencing pipelines, we construct artificial DNA with hidden references for DNA data storage. Noisy reads are aligned to the hidden reference to enable assembly-free and reliable data retrieval. In this work, multiple-fold hidden references enable a fast and reliable readout scheme in a bootstrap manner for large DNA storage, transforming the de novo readout into a resequencing-like workflow. Correlation to the hidden watermark reference identifies low error rate reads, and bitwise consensus generates soft decision information, enabling fast data recovery. The read-by-read forward-backward algorithm corrects indel errors, and alignment to the regenerative reference fills in low-coverage regions, enabling reliable data recovery. To retrieve data from large DNA fragments, conventional methods rely on de novo assembly of noisy reads. These approaches require high sequencing coverage and substantial computational resources, and are further complicated by the diversity of sequencing errors. In this work, we achieved fast bootstrap readout using different filtering schemes based on multiple-fold hidden references. The workflow involves 3 steps: watermark encoding, bootstrap filtering of 3 types of reads, and soft decision decoding. The proposed scheme identifies reads with distinct features in a bootstrap manner. Compared to state-of-the-art methods, our approach achieved low-coverage readout across a wide error rate range. The watermark sequence in noisy reads suffered from interference from the superposed sparsified data and sequencing errors. The proposed method utilized the superimposed watermark sequence as a hidden reference to perform sliding correlation with noisy reads. It enables the effective identification of read positions based on the correlation P2 types of noise. The correlation-based method effectively filters out interfering reads and achieves high classification accuracy. Interfering reads exhibit much lower correlation peaks than data-carrying reads and can therefore be excluded using a correlation threshold. Hidden watermark references facilitate rapid data recovery. Step 1. Sliding correlation to filter low-error reads. Step 2. Bitwise consensus to generate soft information. Step 3. Soft decision decoding to recover the original data. Rapid data recovery was achieved on Illumina sequencing data across 4 code rates at 0.6, 2.5 times coverage, lower than the 20 times typically required by assembly-based methods. This demonstrates the practical advantages of our scheme in reducing sequencing cost. In the presence of indels, we employ the scaffold reference in combination with the forward-backward algorithm to rescue noisy reads. Specifically, a scaffold reference sequence constructed from type I reads is used to partially align correlation failed reads. The forward-backward algorithm then corrects indel errors and generates soft information for decoding. The combination of forward-backward algorithm and the incorporation of type 2 reads progressively improves recovery performance, enabling error-free recovery at 0.8-3.5 times coverage. Since scaffold references often contain gaps, a regenerative reference is constructed from the decoded codeword. Remaining reads are aligned to the regenerative reference to identify type 3 reads for filling low-coverage regions. Finally, a forward-backward algorithm generates a probability-level consensus to facilitate soft decision decoding. For simulated data with an error rate of 1.2%, error-free recovery was achieved at 3.7 times coverage. For nanopore sequencing data with an error rate of 4.7%, error-free recovery was achieved at 4.3 times coverage. These findings confirm that the proposed framework remains valid for low-indel data while offering substantial gains in high-error contexts. In summary, we demonstrate a multi-stage alignment and error correction strategy, transforming the de novo readout into a resequencing-like workflow. Leveraging multiple-fold hidden references, our method enables fast bootstrap and reliable readout for large-scale DNA storage, demonstrated across both Illumina and ONT sequencing platforms.